trial in the human trial, how it affects the antidiabetic properties. So, they use that is a capsule of this bitter gourd, and they can find that there is a significant decrease in the blood glucose level based on this particular trial. And also they have seen that this glucose level increase or decrease because of the decrease in the carbohydrate digestions and absorption. That means your body will not absorb certain amount of glucose to your system, and it will not go to the blood system. So, that is there. Then increase the insulin activities. Okay, it also activates the pancreatic activities. So, pancreas which is giving your insulin. So, that type of pancreatic activity is also increased by this particular crop. So, that is the beauty of this particular crop. Increase glucose utilization, also some of the glucose which is not utilized, also it can also enhance the utilizations by which it can also not come to the bloodstream. So, this is also important that way. Increase insulin sensitive liver and muscles, by which you can have your own metabolic system can be enhanced by taking this crop. So, now there are a lot of studies done in Japan, even in case of Germany, they have institutes which is totally devoted for this particular crop, and having the metabolomites of this particular, which are the secondary metabolites, how it is produced, how it also enhance the health components of the human being. So, that is also they have studied, and lot of findings also nowadays coming with respect to this particular crop. If you see that I mentioned about this one, that how is people of Africa they use this one, because they contain the seed aril, which is very sweet in taste, even just like in sweet taste in case of muskmelon, no? That type of taste will be there if you take. If somebody else can also try, when you go somewhere place and take this bitter gourd, the ripened one, you take that seed aril, which is totally red in color and which is very sweet in taste. I tried. Okay, so don't worry that it will be not having any problem. So you try for that one, which is very important, and people have tried this one. And you know that this person is Simon Groot in Netherlands. They have some of the hybrids which is developed I think way back in 2001, 2002, that is Z Cross. Z Cross is a hybrid which is developed by this East West company. So, it is a very important hybrid, and that time also they have shown that these are the crops which can also give you a nutritious vegetables for and healthy diets. So, this also he emphasized about these crops, those are of now significant, there is a significance is there nowadays for having this health, people are having the nutritious nutritions in the human health. So, that will be very important in this case. So, he got also this World Food Prize laureate in 2019. So, he worked in this particular crop, particularly for this Asiat region. They have some crops, Bangladesh, Thailand, then India, and these people they have tried in some of the crops where they found that this crop is a really potential for different purposes. So, coming to this breeding of this for yield and hardiness, which is very important. If you see the literature, you can find it is not a new crop, it is developed for long back in India itself, and we are said to be the center of origin of also bitter gourd. But nowadays, because of this France, they have claimed that it is not India, it is done by the African is the center of origin. So, we have to again claim our materials that no, it is not like that. We have to have our own materials to be preserved, and I think many of these papers are coming from the African, from the Europe, particularly some people from France, they are working in this particular crop, and they show that Africa is the center of origin, but the center of origin is said to be India, particularly eastern India is said to be the center of origin of this particular crop. So, if you see that crop which is also done by BP Pal, I think he is a rose breeder. Also, he has some of the contributions with respect to this particular crop. In Lufa also he have a very good crop, very good publications. At that time it was in Nature. So, if you say this crop importance at that time, which is also having this published in the Nature during in 1948. Now, these are some of the persons who is really worked. If you go for the cucurbit breeding, if you say the gourd breeding as a whole, these are a lot of like B. Chaudhary is our head of the previous head of this division, also Dr. P.S. Sirai also our head of the divisions in the farmers head of the divisions. Also, if you see this Dr. Harbhajan Singh, he has a lot of contributions to the vegetable is concerned. So, he is also having a lot of materials which is generated by these people, and which some of them are really also going nowadays, like if you say that Pusa domosmi, Pusa vices in bitter gourd still have some good cultivations in India itself. So, if you see that origin and the distributions of this crop, if you say that this crop is totally a diverse crop in nature, and also the choice of the people also different. Some people they need a very long type of fruits, some other places they need a very small type. If you go to West Bengal and other Assam and other, they have a small uche type, we are saying it is a uche type of, it is a very small one and is also very nutritious. If you go to Kerala also, this type of uche type is also very important, they are small type what we call as Momodica charantia variety muricata. Okay. So, that is also very important, and you have also a lot of germplasms where you have white type also there, and if you go to Kerala they need only the white one, like Priya and other, they have their own materials and they got developed this Priya, Priti or something like that, they have their materials are there, and our material is totally different from these other Asian countries. You can see that our is totally in field crop and put in the field, and they have sometimes they need a very strong tubercles having spiny one, but if you go to some other parts they need only the smooth one. If you consider this one is totally a smooth type of like your loofah, okay, like your sponge gourd, you can have this type of materials also we have. And if you go to Vietnam also, these people have this type of particularly for smooth type of bitter gourd they have. So, okay, now is there? Okay, yeah. So, one student also from that part. So, these are the crops, and also what I mean to say, based on the choice of this fruit shape, size, color, everything, our pattern of consuming the bitter gourd is also different. So, you have to breed based on the regional level, you have to breed based on their different characters, which is very important. If you go for only a single trait of this bitter gourd, I do not think nobody will like, but so you have to take the different crops with a different region and different varieties you have to release. So, that is important in case of improvement. If you see the objectives, our first objective is definitely with the yield objectives. It was earlier it was done, but now this changes from the yield to your quality improvement, which is very important. Okay. So, that case you have to go for this quality like fruit quality, anti-diabetic properties, antioxidant properties, then also some of the antidiabetic like saponin, charatin, polypeptides, momodicin, so and so and so. So, a lot of other chemicals which is really important for us. So, you have to breed the varieties in this particular aspect, which is very important nowadays. Then some of the extrinsic fruit quality like shape, size, color, what I mentioned here, it is also very important. Then extended shelf life, sometimes you also have to breed some of the lines, or you have to see the mechanism in which you can just put it that the storage lives will be increased. Then growing season is also important. Nowadays you need to have a off season varieties which can also be grown either by this by this breeding of varieties, or you can develop your own production technology by which you can increase this particular crop. Coming to this species in India itself. So, as I mentioned, there are a lot of species, about 47 species in Africa, and in India we have, including Asia and Australia, we have only 12 species. So, that Scaffer Ranner 2010 is from France. They Spain and France, they are working in this particular crop, and they mentioned about this African origin of this crop, but we say that it is originated in India. But in India, we again, because based on the species diversity there in Africa, they have told that is 47 species are also there, and based on that they are saying it is African in the center of origin, but we say it is in India. And in India we have 7 important species that's grown, and these are of Balsamina, then your Charantia, Dioicus, Hydrica, subspecies Renigena, Cochin chinensis and Cymbalaria. This Momodica Cymbalaria is earlier in the Lupa Cymbalaria, it was in the Lupa Cymbalaria, but again they put it in the Momodica Cymbalaria based on their phylogenetic relationship between this Momodica as well as Lupa. So, they put it there. Still it is in controversial stage whether it should be in the Cymbalaria or it should be in the it is in Momodica or it should be in the Lupa. So, one of our students in Bharati 2010, he worked in this particular species here in our department, and also you can see that Muricata type also we said that it is the ancestral form of this Momodica charantia. Okay, we have one variety that is called as Pusa purbi, which is very small footed type and having dented one, which is utilized for our breeding program itself. Now, if you see this crossability analysis among these species, you can very interesting things you can find it, that Momodica charantia and Balsamina, they have some cross compatibilities there, but it is also again has a lot of problems, difficulties there when you make the crosses. And with respect to Muricata and Charantia, they are very much of crossable each other in both the way. So, if you can say the Muricata and Charantia, they are both the way crossable, but if you see the Balsamina, it is a crossability is there, but it is very difficult to cross. I will go in the details on how it is difficult that to breed between cross between the Balsamina and Charantia. But in other species also, if you see the dioecious species, you can find it that Sahydrica, Dioica, Cochin chinensis and Angulata, and some of them they are both way they are crossable each other, but some of them are having one way crossable, some are also not crossable in the both ways. So, you can see that we have also studied that the cytology of all these species, and also we have seen that some of these chromosomal variations will be there in that species itself. So, that also it will be also having the problem in the crossability. So, that already we have studied, and these are the papers. I can see that the papers which is published in the different journals like Current Science, Grace, Euphytica and Plant Genetic Resources, which is nowadays a lot of people are giving reference in this case. And also we have some paper which is in the Current Science, which is also we have published. Now, coming to that, I mentioned about the Balsamina and Charantia, which is important for us, because if you see this Balsamina, which is also having Momodica balsamina, which is also very important nowadays. The people also taking these particular fruits. It is only in the Gujarat and Maharashtra and some parts of the Punjab they grow this balsamina, and they have one variety is developed by PAU with respect to this balsamina, and they are said to be resistant to red pumpkin beetle, gallfly, pumpkin caterpillar, root knot nematode and CMV, cucumber mosaic virus. So, these are this is the crop, and also we tried in our own field where we put in the net, and we find that balsamina is also resistant to powdery mildew, So, based on this particular crop we tried, and you can see this particular crop small fruited type like Muricata type, but the cost is heavy. If you go to the market and take this particular crop, there is 400 to 500 rupees per kilogram. So, you can imagine this particular crop how is important. And another important is also resistance to different. It is also having the same properties like your bitter gourd, like it is also anti-diabetic properties. People have tried this one, and that is why it is very important this one. Now, if you make the crosses, I mentioned about this, how you can also have interspecific crosses if it is there, how you can develop that some of the crosses, 5 or 10 crosses you take it, but if it is not crossable, then you left it, we are leaving that one, no, we do not need another, it is not crossable. So, report like that. Okay. So, what we did it, we have about 86 accessions of bitter gourd, we cross with the balsamina. Okay. So, all these crosses are not there. Now, there is single seeds and a single fruit you are not getting. Even 1000 crosses is made with the different accession, but we did not get any seeds or any fruit out of that, Now, we have one variety that is Pusa Ausodhi, which is having the crossability with the balsamina. So, you can see when you make a cross in the pre-breeding program, you have to cross many of these species, many of the accessions, okay, then only we can succeed, otherwise do not say that no, it is not crossable. So, when you have that one particular varieties or particular accessions is crossable, you have to tag that one, you have to take it this one for your breeding program, which is important. And now you can see that these are the fruits where there is no normal seed setting is there, at least you can get 2 or 3 or 4 seeds will be there. Okay. So, that is important, and it is there where we are getting here. Then after that we have seen that the analysis, cytological analysis, we did it in the electron microscope, that how this pollen behavior, pollen germinations, how they enter into the pollen sac, and also have self fertilizations, all the things we have studied. Here you can see the important here balsamina with Charantia with respect to the different other crosses, not in the Ausodhi, other crosses we have some there will be a de-link of this pollen germinations here. In this particular point, there is a breaking of the pollen germinations, and it will not, even it is going inside, but it will not fertilized, because there is a breaking of this pollen when you enters into the, and there will be no zygotic formations will be there. But again also there is formations of the callus. Okay. So, these things will be there, and there will be no seed setting or fruit setting will be there. But interestingly, in case of Charantia with respect to this your that is Ausodhi, we crossed about 7,100 flowers, but we got also many seeds, about 5,000, number of fruits also get a huge amount of fruits you can get it. Whatever amount, just like a cross of your selfing of your Mammodica charantia, you can get the cross fruits, and we have obtained the seeds of this much, but only 113 seeds are good. That means the good quality seeds will be very, very less if you compare to when you go for crossing with all the seeds together. So, that huge crosses you have to take, and 110 seeds, 13 seeds only get out of this one. And again, the plants we generated, only one plants we got it from here. So, it is the only one plant when you got it from there. So, you can see the imagine the 7,800 and you have 5,000 fruits, you have 88,000 seeds, only one seeds you are getting, which is having your this one, giving your F1 seeds. So, that type of work if you continue, then only you can be successful in any breeding program. So, this is the cross and this is the F1, and we also see that morphologically some changes is there, but most of the cases, most of the traits are similar to the female plant, like also the. In the F1 itself, most of the traits we imagine the morphological traits, we find all are from the your female plants, and very, very less traits which is like your tubercles, it is a pointed one, here it is also having pointed or spiny one, but other than that this is not having this one, other fruit shape, size, everything will be just your female one. So, that also you can see it. Then after that what happens, so once we get that one, we just go for crossing with this one, with this both the way. We cross with this charantia, that is also the again, also we cross with the Mammodica balsamina, because at that time we thought that this is always behaving like a also the. So, if you have some of the plants, again we cross with this balsamina, then only you can make some success. So, where we did it, we go for back crossing, we cross with again with the balsamina with that particular F1. Okay? We cross with this balsamina and we got some of the back cross population, and out of back these populations we got only 3 plants. But among these 3 plants in the back cross population, you can find that all are chlorotic. Okay? All are chlorotic. That means once the genes from that balsamina is coming to this your background for charantia, it having deleterious effect and there is no survival of the plant. Then we take that DNA of this particular 3 plants, and we have also seen there is an interrogations of the balsamina genotypes in our lines. So, now I think we have to proceed here. We can find that you have to go for more and more back cross, more back cross you can generate more of seeds, may you can get it something there with this one. Again we tried with different other methods for interspecific hybridations. We cut that the seeds particularly for the embryonic region. We did lot of experiment on this particular field, but only this much we can get it from here. But if you again go for crossing with back cross with this one, but we are thinking that we can also get some of the lines from this particular lines, if it is there, when in the future cross program we can succeed this one. Another program we have taken this gynoecious, that is predominant gynoecious line, which is wonderful in case of a particular crop. Nowadays many of the people, many of the universities claiming they have the gynoecious line, but the origin may be of same or maybe of different, but the thing is that people are nowadays getting a lot of gynoecious line because as you know there is a difficulty, it is a small flowers, there is large number of flowers, male flowers are there, very less number of female flowers will be there in any particular species, a monoecious in nature. So, based on that we have got some of the lines that is from Jharkhand, we have 2 lines, 2 seeds we got it, you can say that, which is having gynoecious in nature. We plant them in 2006. One of my student SS Dev, who is I think he has taken the classes here. We in the field itself we got 2 plants out of thousands of plants we grew them, different lines we grew them, we just collected from NBBGR, and also we went to Jharkhand and other states for having the germplasm collections, and we got it only 2 plants. So, surprisingly this when you go for these 2 plants, you have having the gynoecious in the 2 plants, and they are very small size in about 4 to 5 centimeter long length. And they again what happens when you go for these 2 plants to maintain them with this silver thiosulfate and silver nitrate, the plants dies. So, before that what we did it, we take the shoot plant and go for tissue culture. So, another method we adopted, immediately we adopted that one. One of my students Swati did it this one. So, she did it the tissue culture, and we generate about 3000 to 4000 plants with respect to tissue culture, and then we plan our methods having inducting the female flowers in this particular crop. Then they have tried and we got it some hermaphrodite flowers. Okay, so, that already I mentioned about the hermaphrodite flowers that we got using the silver thiosulfate. One example is telling that in case of bitter gourd and cucumber, you have to spray them, that also we will discuss that whether you have to spray them earlier in the 2 to 4 stable stage or any stage of the crop plant, you can also have the spraying of this crop. Now, what happens in the when you go for this one, we also have some of the lines or some of the plants based on these different spray combinations like silver thiosulfate, silver 6 millimolar, 3 millimolar, GA3 nitrate and nitrate. We are only succeeded in these two concentration, that is 3 millimolar and 6 millimolar, but 6 millimolar is toxic to the plant. So, if you go for 3 millimolar, it will be definitely giving the hermaphrodite flowers. So, what happens when you go for this developing the hermaphrodite flowers in this gynoecious line, again there will be lot of things will come in the picture, because what happens if you go and spray them and you spray in the first spray any stage of the plant growth, you have to again after 7 to 8 days again you have to spray them with silver thiosulfate, either 3 molar or millimolar or 6 millimolar. But what happens when you go for that particular hermaphrodite flowers, you self them, you will not get any seeds, another problem will be there. So, that time also we did the same things, we got it there. There is no seed setting when you go self them, because pollens are there, but there is no seed set, because when you spray them there is stigmatic surface will break, bifurcated. So, you will have a pollens, it will not germinate there, it will not go to the for fertilizations. So, what you have to do, you have to wait another 10 days without any spraying, you wait another 10 days, the female flower also will come, hermaphrodite and the female comes simultaneously. Now, what you will do it, you take that hermaphrodite pollens, put it in their female flower, you will have a 100% seed setting. So, this is the peculiarity. So, many of the people have tried, but they have not till today having this powerful technology, but they do not have. Now, they do not have the inbreds, they are behaving like just like in monoecious, some are having gynoecious, and they do not have this type of lines which is totally inbreds and giving 100% female flowers. So, whenever I show that one to the people, they say okay, we do not have a gynoecious, but how to maintain them, we have the same problem coming. So, that is the things you have to, if you go for maintaining that, you have to have some of these clues. So, we did it that one. Now, after getting this one, we tried with some of the things like you know that MCS2 gene in case of cucumber, CM2, CM7 up to that 7 genes are there, which is really giving you to the femaleness and your insulin production genes, or which is very important. So, we tried with this one that how in our bitter gourd how these genes is acting. Okay. The same genes having SCS2 genes, which is very important particularly for ethylene evolution genes, and which is also we tried it there. We did the same thing here, we go for before spraying we take the buds. Okay. That is because flower development also changes with respect to these particular genes when you have a different primordials coming in the play. So, how it giving to the flowering hormones, which is very important for this particular having hermaphrodite or the your female flowers will be there. So, in that case we go for spraying, and it is a control. Also we spray up 3 days after first spray again in the T1, 3 days after second spray, that is second spray after 7 days of interval we did it, and also at the conversion stage when this female flower converted to the hermaphrodite flowers, you take the different things and that bud samples, and you analyze them with this PCR with the expressions of the genes. We analyze the ethylene evolutions through the gas chromatography. Also, we use that GO3 with the HPLC. So, there are a lot of things we did it in this particular expressions of the genes, and we can see that this is the genes also. If you see the ethylene concentration in monoecious is very less compared to the gynoecious plants. Okay, and if you see that as monoecious it will be less compared to the gynoecious, the ethylene production, and if you see the expression level at the different stages. Okay, and you can see that in the control where you have not sprayed any silver thiosulfate, you have a very good expressions of these SCS2 genes, and it is also reductions will be there when you go for treating this your silver thiosulfate. So, this says when there is an ethylene production in the female production, but if you spray with the silver thiosulfate, it reduces the ethylene perceptions, and there is less accumulations of the SES-MRR gene, which suppresses the productions of the ethylene. So, in the ethylene productions, when it suppresses along with this endogenous level of GA, when it will be optimum conditions, it gives to the hermaphrodite flowers. But otherwise, if it is not suppressed, it will be only giving the ethylene production, it will be more of your femaleness, you will not get any hermaphrodite flowers. So, this is the things what we generated with respect to this particular gene, particular in this case of SES2 gene. Now, coming to that background of that, what we did it in the female flowers, that is a small fruited type, but people need to have a longer fruited type or the intermediate length of the fruits, maybe 10 or 15 centimeter long will be sufficient. If you go, they need to have this particular consumer preference will be there. If you go only in the eastern part, you have a small fruited, but if you go to the northern part, you go to south part, they need a longer fruited type, maybe in intermediate type fruits. So, we just study the genetics of this particular trait of the fruit length, and you can find the small one that is there, and we have a long one about 35. You can see the full length of the scale. You can see that particular variety will be is there with us S2 lines. So, we cross them, and we also plant for this genetics of this one, and we studied that this particularly there is 4 to 5 genes involved in this length, and the small fruited type is incompletely dominant over the long fruited type. It is there, and there will be 4 to 5 genes will be there. So, that is the study what we got it from here based on this particular aspect, and then we used that one for our breeding program. So, we got it in 2006 that particular small lines, we make a cross with the traditional breeding. You can see how much we did it here, and it took about 8 to 10 years for getting a good lines with having the intermediate type, having the gynoecious line. That time we do not have molecular markers approach, and that time we only that genome sequencing is not available in beta gourd. So, we could not carry out this that also. So, we did the normal breeding because it is a gynoecious which is in also recessive gene. So, we make a cross, then selfing them, again evaluate them for the phenotypic evaluate them, then you take a cross with them again with this one, a back cross populations we developed, and simultaneously you have to every back cross populations you have to just take some of the lines which is having the more the fruit length will be at least 15 centimeter long. So, there you select that particular lines, and up to 6 generation we maintain them, and after 6 generations we have that lines having the gynoecious nature and the characters of Pusa biseus and Pusa domosimi. So, this is the thing. So, here we have in this particular populations we have 2 types of population we developed, only single cross with a different back cross we got 2 populations. One is having the tubercles, another not having the tubercles or cylindrical type. So, we put it as a GY23-1, another is your PVGY201 or what we have GY1. So, these are the 2 lines developed after 8 to 10 years of your back crossing. Then another problem is there, as I mentioned that in case of gynoecious line it will be very difficult to maintain, a farmers cannot maintain, you cannot also maintain. So, that is why we need to have a predominantly gynoecious line. Here we do with this predominantly gynoecious means all the time it is having female flower, but in the latter part of the plant you have only hardly 5 to 6 male flowers will come. Okay? So, that way if you have a latter part you can also take it this one and make a maintain that particular line. So, that is why we have one predominant line gynoecious lines also developed with the same populations. We have this particular predominant gynoecious line, and you can see this predominant gynoecious line in the background of Pusa domosimi, which is having a number of female flowers will be more, and we also registered in the NBBGR. Okay. So, this is the lines we have called as pre-G1, we put it as pre-GY1 and which is registered, and you can see this beta gourd where you have a number of male flowers are more, but here you can see the number of female flowers will be more, but in the last phase of your plant you can get the male flowers which you can utilize it even for the maintenance of this predominant gynoecious line. So, this is there in case of beta gourd. Then we developed some of the varieties like Osodhi and also Pusa burbi, which is also in the trial. Also, we developed some monoecious hybrid like Pusa hybrid 2 and Pusa hybrid 4, which is released also in the national level as well as the state level, and which is also utilized. In the meantime, also we just generate some of the molecular breeding approach, particular Marcus breeding approach, and I think this is the first paper, this was genetic diversity in beta gourd with RFPD markers and agronomic tests. You can find in 2006 by one of the students that he worked in this particular aspect. At that time, it was the first paper in this bitter gourd having this diversity study with respect to this RFPD that time we were doing. So, he did it that one. Nowadays, a lot of papers are coming in this respect, not only RFPD, not a lot of other markers that people have evaluated in this particular field. So, then these are the publications we also generated, and also this one I mentioned that how it having the gene expression studies already the last time we also shown this one. Then we validate markers and we can find some of the markers, and also based on this genomic library we developed some of the markers, and this is another important work which we did it. Also after that genome sequencing is available nowadays. So, we also did this Gangadhar, my students also work at this particular aspect, and we also have some SNPs which is mostly utilized at QTL's mapping, we did it in this one, and also we have some of the genes we also find it in GY1 also markers SNPs which is linked to this particular trait. Now, again another traits we also found that some of the important economical trait like your fruit tubercles, which is very important, and this tubercles is always dominant over the smooth one. So, this is the smooth one, we have lines, that is we have Pusa rasdar which is having smooth one, and we have your Pusa purbi which is having the tubercles one. So, we make the crosses and you can find some of the genetics of this particular traits, and again based on this SSR available in our system and the public domain, we just screen them and we found that some of the markers is linked to, it is not so much of tightly linked, we can go for fine mapping of this particular crop, then only you can go for linking of these markers, otherwise you go for SNPs or some studies by which you can also develop the markers. But still we have some SSR which is also linked to this particular traits. Again we see that with this association 1, we have seen that how this marker or this marker which is also showing this similar type of pattern when it is having this either in the tubercle soil or in the cylindrical. Okay. So, this particular base pair of DNA also similar to in case of your tubercle type, and this particular base pair is similar to your smooth type. So, they also develop, but we need more confirmations, it is only the preliminary work, and only because the distance is very large. So, if you have some markers which is tightly linked, then only you can have a very good one. Now, coming to this ridginess is also very important. I mentioned that some are having discontinuous ridges, some are having continuous ridges, and discontinuous ridges is incomplete dominant over the ridge one. Okay. So, that also it will studied, and also we have seen some of the markers which is flanked to the gene of interest about 19.6 centimeter distance apart. So, that also we can also find it here, and which can also be utilized. These are the some preliminary work we did it, but I think we have to go for more and more with respect to these genes and their positions. Once the linkage map will develop, definitely we will have some sort of program, and when we go for resequencing of the different materials, we can also have the different type of things we can get it. So, this is our future work what I see. Now, breeding for the protected cultivations, at that time you know that if you grow only cucumber and your tomato in case of your protected cultivations, we thought that some of the lines we can also take it from these different collections throughout the world, and we make it from the Asian collections, and some of the hybrids are having very good result in this case. So, we break them, we just develop that particular lines from that one which can be utilized by the farmers, the small farmers they can also develop this one. So, we have some of the lines like here I mentioned about 4 lines which is really promoting to this polyhouse cultivations. You cannot grow in the outside. If you grow them outside, there will be no fruit setting, and the plants will remain only hardly about 1 feet long. And if you put in this net house or polyhouse, it will have very vigorous growth and you have a very good fruiting will be there. So, that we developed this one. So, here you can see that particular Pusa rasdar which is having capsicum in shape and also having very good juice content. It is rasdar means it is having juice. So, high amount of juice is content, but because now the people prefer to have the juicy one to have their at least for drinking purpose in the morning, they need to have a juicy one and less bitter compared to our old Indian lines. So, because if it is more bitter, you cannot take the juice in the raw. So, that is why we have the lines which is Pusa rasdar. I think we put it in the Bihar, that is Sabor, no? So, that lines, and you have taken that one. But thing is that we have to commercialize them. It is very important because as a health conscious people are nowadays coming, and if you make a juice out of that, it will be very important for us. So, and another important thing is that it is also predominantly gynoecious line. It is also predominantly gynoecious line, and but you have to make selfing yourself. It is the only constant there. It is not parthenocarpic. You have to make selfing, but one fruit is about 100 to 200 grams of fruit weight. So, there is no problem if you get selfing, and within a 100 square meter of area you can get about 4.7 quintal of fruits. So, these are huge productions will be there, and the cultivations are good. Definitely this type of fruit productions you can get only 100 square meter polyhouse or net house. So, that you can do it. We also evaluated them in the CPCT. We got a very good result with respect to this Pusa Rasdar. We also released that one. You can see the size of the fruits here. So, this is totally, you can from the beginning to end you can have these fruits of this much of, and once you, because as you know that you have to harvest every time. If you are blocking the harvest, you will not get the new fruits. That is usually in cucurbits is very important. So, at least in a one bunch one time we can have 4 to 5, 6 fruits you can get it. If you harvest continuously, definitely you have a very good yield in this particular crop. So, we have a different lines also is concerned to this one, but this is the white lines and having very you can see the crocodile type of this one, the shape of the fruits, and also the skin of the fruits, which is also very important. But only drawback we have here is that is a whitish type fruits, white type plant. Another things you can also develop here, that white plant fruits, those are having white, except Indian like your that Kerala variety Priya. This is having this variety which is shy bearer. Many of these white fruited type in bitter gourds are shy bearer. So, if you compare this one, go for genetic studies, then only you can evaluate or something we can find it. And also it is very late bearing, very late. Okay. So, even after 3 to 4, after 80 days of your plant, you can also get 1 or 2 fruits. So, that type of genetics also you can study that how it is be there, why it is so like that. So, that type of things also you can work out here. Then what we did it, we have a plan of studying these varieties of the gynoecious line. We give it to 2 companies, the private company, one is on Kuru and Namdari. We also get 3 lakhs rupees from them, and generally what happens, the gynoecious line they can take it and they go for hybrid seed productions, and easy for this one because you do not have to go for pinching other things, because pinching or hybrid hand pollinations will require lot of money. So, if you have a gynoecious line, grow them and go for the pollinations, you will have a automatically you can get these hybrid seeds. So, that is very important. So, they license it, and now they are having very good satisfied with this particular lines. But these lines is really I am telling it is because this is having a stable lines. Other lines the people are developed, but they are not stable because they are not the inbreds. So, inbreds if it is there only will be stable, other will be not having not stable. So, then we have some of the other companies we are giving this one. The some small of the things I mentioned here about this quality aspects. Quality is very important, and we also have the variations in the quality aspect. If you see this because we did have some of the program with the National Institute of Nutrition Hyderabad. So, they evaluated our germplasms with respect to the different quality attributes coming from the minerals to antioxidants and vitamins, and you have a lot of variations will be there. I will take some few time here. So, these variations will be there. Now, another important thing is that I already mentioned in the growth stage, how this type of anti-diabetic properties as well as antioxidant properties are changing with respect to the fruiting stage and the flower ripening stage. So, that also we have evaluated. We have seen the different stages of that, how it is totally changing, and also we studied the expressions of some of the genes, particularly for carotenoid biosynthesis genes with respect to these different materials what we have, and with respect to Pusa Rasdar, which is grown in the protected cultivations, as well as Pusa VCS, which is grown in the open field condition. So, we studied that gene expression studies with respect to these 2 varieties with respect to different days of your anthesis. After that we have studied that one, and you can find a lot of changes with respect to these particular genes is concerned. All the 3 genes having the increase with respect to this when you go for ripening of this particular crop. So, enhancing these lines also, particularly bitter gourd, as you mentioned about Pusa Rasdar, which is you can if you 1 day or 2 days you can put it in the outside the room temperature, it becomes shrinking, because I told it is a very juicy content, but once you put it in the room temperature it becomes sprinkled. Okay. So, public will not take this one, they will not take this particular. So, we have to go for some of the technology by which we can also increase the storage life of the shelf life. So, we have some of the gum coated type just like your guar gum. So, with that latex you can take it and you can also make it this one. So, we have a advantage about 6-7 days or 7-8 days of your storage life we can enhance this one. So, this is the already we released this one in front of you, that particular bitter gourd genome. So, we have published it this year only 2020, and if you see that strategy of the breeding, we must have to have lot of strategy. As I mentioned, in a single crop you work for a whole life, you have a lot of things to do. Do not go for thousands of crops. I do not think only single crop you take, you have a lot of chance to have all the things or detailed study you can do it. It is not like rice or wheat, many people are working. Even a single person working in the single crop, you can also develop a lot of lines in your future. So, that is the message what we say. So, I must thank to my old students, those are really a good worker to me and having a good background of their work. So, I published lot of paper, lot of work in bitter gourd, and also the mentors, so which is really important for me. Also, I have some people working with me, Dr. Munshi, Ranjit Ranjan, Ambika, and Gograj is also here, and my technical staffs, and I have also very good projects in DBT, ICAR-IARI, this is there, and PPV which really contributed to me. So, these are some of the most important publications which you can refer to you in bitter gourd. You can have this type of publications. If you do it, I think it will be wonderful for you. And this is I visited your open field day World Vegetable Center in Thailand. Huh? I went to Thailand, Bangkok. So, that is I told. So, this in the Bangkok is a bitter gourd germplasm is there. So, here this is Taiwan, but it is in Bangkok, Thailand. Okay. So, I went given a lecture there. So, these are huge collections, they have hybrids in bitter gourd and dimensions. So, thank you very much. Thank you very much. So, if you have any questions and if you worked in this particular crop, I will be most of it will be appreciation for you. Thank you. Any question, anything?